Hello guys, welcome to BioTube. Today we are going to learn about cell cycle and cell division. So, what is cell cycle? Cell cycle is the sequence of events by which a cell duplicate its genome, that is nucleic acids, DNA, RNA, synthesizes other cellular constituents, that is the organelles like mitochondria, Golgi body, ER, etc., and finally divide into daughter cell. So, normally, what do we observe? We observe that every organism has come from a single celled zygote. So, how does a single cell zygote form such a complex organism like us? Okay. Obviously, the zygote undergoes cell division because in our body we have millions of cells, no? So, how does this happen? This happens because obviously the zygote has undergone some divisions. Okay, otherwise, how is it possible? Obviously, the single cell zygote have to undergo cell division in order to produce uh, millions of cells. Okay, so we can conclude about two important events, that is cell division and growth. So, why growth is important? Suppose this is a very big room and we divided it into a number of compartments. Then in each unit the surface area is very much small. So what will happen? The cell will divide, it's okay, but ultimately the surface area will be very less. So what will happen if each unit divides independently? Each unit here divides independently. So the area will obviously increase. In this case, growth is important to increase the surface area in order to maintain the nuclear cytoplasmic ratio. Okay, let's see about the feature of cell division. Cell division is a sequential and coordinated process. ऐसा नहीं है कि पहले DNA replication होगा then growth होगा वगैरह वगैरह नहीं है. First there will be growth, then there will be DNA replication, and then there will be cell division. It's a very sequential and coordinated process. Okay. सेल डिविजन ऑकर इन बोथ प्रोकैरियोट्स एंड यूकैरियोट्स, मतलब बोथ प्रोकैरियोट्स और यूकैरियोट्स में सेल डिविजन होता है, बट एज इट इज़ नॉट प्रॉपरली विजिबल इन प्रोकैरियोट्स. प्रोकैरियोट्स में ऐसा सेल डिविजन बहुत ज़्यादा विजिबल नहीं होता क्योंकि इसमें प्रोकैरियोट्स तो प्रिमिटिव है ना, तो सब ऑर्गेनेल इज एब्सेंट है, so there is not proper nucleus is not properly developed. So because of these issues, cell division is not properly visible in eukaryotes. So we consider that cell division is a feature of eukaryotes, but we have to always keep in mind that in both prokaryotes and eukaryotes cell division does occur. Let's come about the phases of cell cycle. So cell cycle, we can divide it into two phases, that is cell division and interphase. Cell division means it's the actual phase when the cell undergoes division. मतलब ये वही टाइम होता है जिसमें सेल का डिवीजन होता है. और इंटरफेस टाइम पीरियड बिटवीन एंड ऑफ वन डिवीजन एंड बिगिनिंग ऑफ द नेक्स्ट साइकिल. So we can consider it as a resting phase. सपोज आपका सेल डिविजन स्टार्ट हुआ है मॉर्निंग 7 AM में, ओके, और एंड हुआ है लेट सपोज 12 PM में. देन सपोज आपका वन सेल डिविजन 12 PM में एंड हुआ है और आपका नेक्स्ट सेल डिविजन स्टार्ट होगा 9 PM में. Okay, so the gap between 12 PM and 9 PM, that is regarded as interphase, that is the resting period. In this phase the cell will prepare itself for division. Okay, मतलब ऐसा सोचो कि हम किसी ट्रिप में जाने के लिए तो पहले प्रिपेयर होना पड़ता है ना। पैकेजिंग करना पड़ता है। और सोचो कि तुम्हारे कल एग्ज़ाम्स है। सो एग्ज़ाम्स के लिए तुम तो जाकर दे नहीं पाओगे ना विदाउट प्रिपरेशन। सो एग्ज़ाम्स के लिए भी प्रिपरेशन करना पड़ता है। सो एवरी मेजर प्रोसेस, इन ऑर्डर टू डू एवरी मेजर प्रोसेसेस we have to prepare ourselves, no? So cell also prepares itself for division. So interphase is divided into three subphases, that is G1 phase, S phase and G2 phase. Okay, so G1 phase, in this phase cytoplasmic growth occurs. Okay, organelles, that is ER, Golgi body, etc. doubles. Transcription translation occurs, very important point. Transcription and translation occurs. Why transcription and translation? This is because सेल डिवीजन के लिए हमें एंजाइम बहुत ज़्यादा रिक्वायर होते हैं. ओके, सो व्हाट्स अबाउट द केमिकल नेचर ऑफ एंजाइम्स? Obviously enzyme is protein. Okay, so first we have to undergo the DNA has to undergo replication in order to make the RNA, and finally the RNA will make the proteins, no? So in order to according to central dogma, in order to supply the enzymes, the transcription and translation process must have to occur. So G1 depends on external nutrition, and so it's variable. Sometimes G1 cell cycle is variable in different organisms. This is because of G1 only, because the G1 phase depends on nutrition. Suppose if you are not taking adequate nutrition, the cell division will not occur properly. After G1, three options left for cell. G1 के बाद सेल के पास तीन ही ऑप्शंस बचा होता है. एक कंटिन्यू सेल साइकिल और एंटर एस फेज. आइदर इट सेल साइकिल को कंटिन्यू कर सकता है और एग्जिट सेल साइकिल एंड एंटर रेस्टिंग फेज. और आइदर ऑप्शन 2, इट कैन एग्जिट सेल साइकिल को छोड़कर वो रेस्टिंग फेज में जा सकता है. और सेल नॉट कंटिन्यू टू सेल साइकिल एंड बिकम्स परमानेंट. और ऑप्शन नंबर 3, दैट इज द लास्ट ऑप्शन, इट कैन बिकम परमानेंट. ओके, so let's see what if the cell enters the S phase. So what happens in S phase? S phase stands for the synthesis phase. So what is synthesized in S phase? Here the DNA gets doubled by the help of the enzyme DNA polymerase. We shall learn about DNA polymerase, DNA, RNA, genetic material in our subsequent videos on genetics. Okay, then comes the G2 phase. Here the cell growth continue. Transcription plus translation continues. Okay. Let's see about animal cell. Animal cell में एक यूनिक फीचर होता है centriole. These are minute cylindrical organelles. These centrioles are required to make the spindle fiber or spindle apparatus. The spindle apparatus separates the sister chromatids between daughter cells. We shall learn about them in the subsequent videos. Let's see the duration of human cell cycle. So human cell cycle the duration of human cell cycle is 24 hours, and it is subdivided into M phase, G1 phase, S phase, and finally G2 phase. Okay, M phase lasts for 1 hour, that is it constitutes only 5% of the cell cycle. G1 phase continues for 8 to 12 hours, that is it constitutes about 33 to 50% of the cell cycle. S phase constitutes for 6 to 8 hours, that is 23 to 25% of the constitution of the cell cycle. And G2 phase lasts for 3 to 4 hours, that is it constitutes 15% of the cell cycle. So let's summarize what we have learned today. So we have learned about cell cycle. Cell cycle means the sequence of events by which cell duplicates its genome, synthesizes other cellular constituents, and finally divides into daughter cells. So we have observed a single cell zygote undergoes division in order to produce a complex organism like human beings, okay, by cell cycle and growth. So we can conclude about two important events of cell cycle, that is cell division and growth. Growth is required in order to increase the surface area, because if there is cell division without growth, the nuclear cytoplasmic ratio will change. In order to increase the surface area, growth is very much important. Then we have learned about the feature of cell division means it's a sequential event. It occurs in a very coordinated way, and it's a feature of both prokaryotes and eukaryotes. But it is not properly visible in prokaryotes, so it is considered that cell division is a feature of eukaryotes only. Then we then come to the phases of cell cycle. Cell cycle is divided into cell division and interphase. Cell division is the actual phase in which cell undergoes division, and interphase is the time period between end of first division and beginning of next cycle. That is, it is the resting phase. It is further subdivided into G1, S, and G2 phases. G1 phases, here the cytoplasmic growth continues, organelles doubles, transcription translation occurs, and G1 phase depends on external nutrition. Okay, S phase, here DNA doubles, enzyme DNA polymerase helps in the duplication of the DNA, and G2 phase cell growth continues, transcription translation occurs. There are some unique features in animal cell, that is present of centriole, which helps in cell division by the formation of spindle fibers. Okay, duration of human cell cycle is 24 hours. It is subdivided into M phase, G1 phase, S phase, G2 phase. That is 1 hour, 8 to 12 hours, 6 to 8 hours, and 3 to 4 hours. So that's all about today. Thank you.